Let's move to the next topic, monohybrid cross. Before learning monohybrid cross, can you identify Mendel's gene which regulating color of pea plants, that is white color in pea plants? The mystery of regulating white color is solved. Now, let's see what are the characters Mendel chose. There are 7 different characters. These 7 characters in garden peas shown on the plant's 7 different chromosomes. These are 7 different chromosomes, 1, 2, 3, 4, 5, 6 and 7 chromosomes. All these 7 chromosomes which shown the 7 different characters. 7 characters in garden pea plants are shown in 7 different chromosomes. What are the characters? The 7 characters are flower color. Color of the flower. There are 2 colors, purple and white. Color of the flower. Seed color, yellow and green. Pod shape, shape of the pod, inflated, constricted. Position of the flower, axial position or terminal position. Height of the plant, tall and dwarf. Color of the pod, yellow and green, and shape of the seed, round or wrinkled. So these are the 7 characters, contrasting characters which are shown in different chromosomes. So now these characters which are called as genes. So here we can say as factor or determiner or trait. So these are the 7 contrasting characters. Before entering into monohybrid cross, let us see how this white color flower, what is responsible for regulating white color flower. In the year 2010, there were a group of scientists, geneticists. They discovered there is a gene which regulating the color of white. So it's quite fascinating to trace out Mendel's gene. This name of the gene is P gene A. The name of the gene which represent the color of the flower is P gene A, which encodes a protein. The P gene A, name of the gene is P gene A, that encodes a protein that functions the transcription factor. Transcription factor. This transcription factor, it functions as transcription factor that responsible for the production of a pigment anthocyanin. The name of the gene is P gene A, which encodes a protein. The name of the protein is the protein that functions as transcription factor responsible for producing the pigment. The name of the pigment is anthocyanin. If the flower which consists of the pigment anthocyanin, the flower color is purple. The color of the flower, there are 2, purple color and white color. If the production of anthocyanin pigment, the color of the flower is purple. And this white color flower do not produce this anthocyanin pigment. So the researchers, the team of researchers, they introduce by gene gun method into this anthocyanin pigment, this P gene A, introduce the P gene A into the petals of white color flower. When this P gene A introduced as a single sequence, it changed the cell and this produces anthocyanin pigment and it becomes purple color. But what about the white color flowers? In white color, it inactivates the transcription factor. So there is a mutation occurs in this, and due to the nucleotide change, there is a single nucleotide change. So the single nucleotide change which stops this transcription, it inactive. The transcription factor will be inactive, so there is no production of anthocyanin pigment. If anthocyanin pigment is produced, it will become purple color. The flower color is purple. When it is inactive, this transcription factor, when it is inactive, it do not produce anthocyanin, so it become white color. So the mystery of this white color can be solved now. Now let us see monohybrid cross. Monohybrid cross or monohybrid inheritance is the inheritance which consists of a single trait or single character, that is height of plant. Height of the plant. There are single character, the height, the parent one is tall, is crossed with dwarf. So there is the character is height of the plant. In monohybrid cross, he selected only one character, that is height, tall plant and dwarf plant. So when this tall and dwarf in the F1 generation, so when you make a cross between tall and dwarf, it produces two generations, F1 as well as F2. In F1 generation, this all produces, the alleles are capital T, small t, small t. So this allele, two different alleles with single gene. So this alleles consist of T, the gene T, here small t. This T is a gamete. This T is the gamete. These are alleles. Alleles are represented in pairs. So this gives all tall in this capital T, small t in F1 generation, which produces all plants are tall. So when he experimented 1064 species out of 1064 in F1 generation, all will be tall, and the dwarf is not expressed in F1, but it is expressed in the F2 generation. So in F2 generation, it gives the ratio 3:1 phenotypically and 1:2:1 in genotypically. So out of 1064, 787 are tall in F2 generation and 277 dwarf in the F2 generation. So F1 generation all plants were tall, but F2 generation it gives 787 tall and 277 dwarf out of 1064 species. Let us see how it will be. There are two words which named as genotypic, genotype and phenotype. Genotype is the genetic constitutions. Genetic constitution is called genotype. Phenotype is the observable characters. These all characters are observable characters. When among this 1064 species, the height it is expressed, observable character, tall or dwarf, or this seed color of the seed, observable character. But genetic constitution is called as genotype. So when it is calculated, the probability of this, it gives the ratio 3:1 phenotypically. Let us see how it is identified. So this genetic constitutions are easily represented by a diagrammatic representation, or it is calculated in the form of a checkerboard, or it is named as Punnett square. Punnett square, after the name of Reginald Punnett, it is called as Punnett square. So it illustrated very easily this probability of this ratios. the identification of this ratio. Very easily it can be calculated. Let's see. The parent plant is parent, capital T and small t. This is pure breeding variety, true breeding tall and true breeding dwarf. Tall and dwarf is the parent. So in F1 generation, all these plants, the tall and dwarf, when it is make a cross, it produces all tall plants. How it produces tall? This is the Punnett square. Here in F1 generation, this is the F1 generation. One of the parent is capital T, another one small t. So it produces here this and this, capital T, small t, this and this, capital T, small t, this and this, capital T, small t, this and this, capital T, small t. So every offspring is tall, capital T, if the allele which contains dominant and recessive, that is capital T and small t. In the first generation, it expresses tall. So here it is tall, this is also tall, this is tall, and this is tall. Every offspring is tall in F1 generation. But in the F2 generation, the F1 generation can be selfed. So this capital T, small t is the F1 offspring, can be self cross. This is self cross, hybrid with hybrid. So when a hybrid is, these two are pure breeding, capital T and small t is pure breeding, true breeding parent, but this is hybrid. So the hybrid of F1 generation can be selfed. When it is self cross, it produces in the F2 generation. Let's see how it gives the ratio in F2. Here one of the parent is capital T, small t, another one capital T, small t. Let's find the first offspring. It gives capital T. Here capital T and small t is there. So dominant character, let's write first capital T, small t. Here capital T, small t, and here is small t. So here this is capital T, both are dominant, the alleles are dominant, and it produces tall. Here and here two alleles which represent capital T and small t. So capital T is dominant over small t, so this also tall, and this is recessive, small t, so it is dwarf. In F2 generation, in F1 generation dwarf is not expressed, but F2 generation dwarf also expressed. In F1, this shows the F1 generation. So this is the Punnett square, which is the diagrammatic representation of this ratios. So very easily we can find out when it is shown in the Punnett square. So this is the F1 generation, all plants were tall, but here tall and dwarf. Now the ratio here, capital T, and capital T, small t, and small t. So it gives the ratio phenotypically. As I mentioned earlier, phenotypic is the observable character. So phenotypically it produces 1, 2, 3 tall, 3 is to only 1 dwarf. So phenotypic ratio, monohybrid phenotypic ratio is 3 is to 1. But genotypic ratio is, there is one capital T, 1 is to 1 capital T, small t. Here 1, 2 capital T, small t, 2 is to 1 small t. So this is homozygous tall, this is heterozygous tall and this is homozygous dwarf. So, the genotypic ratio is 1:2:1. This is monohybrid cross. So, you understand that monohybrid cross, there is a single character. Here the character is height of the plant. F1 generation, all plants shown tall. But F2 generation, it gives two different ratios, phenotypic as well as genotypic. Phenotypic ratio is 3:1 and genotypic ratio is 1:2:1. Then another term is there, reciprocal cross. Reciprocal cross. What does it mean? Reciprocal cross. In one experiment, we are taking this capital T as a capital T crossed with small t and the small t into capital T. So, both the results are same. That is, so reciprocal. If you take this capital T as male and this small t into female, or this capital T female and this as male, the result is same. So, monohybrid cross, Mendel's monohybrid cross, so that it is, for example, if you are taking the pollens of capital T, tall plants pollen dusted on dwarf plant stigma or dwarf plant pollen into tall plant stigma, the result is same. So that they conclude that Mendel's monohybrid cross is not sex determined. So, one word question it may ask, Mendel's monohybrid cross is not sex dependent due to the reciprocal cross. Reciprocal cross, it gives the same ratio. So, it gives the same result. I hope you understand this clear. So, what you learn once we recall. Monohybrid cross, very important question. Monohybrid cross, first learn what is monohybrid, what are the characters are selected. It is a single character selected and draw this Punnett square. Now you have the study material in your hand. So, you can easily learn this question. And these are the characters which is represented in different chromosomes. And how this, how the white color flowers, how the gene is responsible, the mystery of white color flower is solved according to Mendel. So, learn this question before the next class. Thank you.